This novel ocular translaminar autonomous system uses the human posterior segment to independently regulate intraocular and intracranial pressures to generate a translaminar pressure gradient. The TAS model allows the evaluation of human intracranial pressure in an ex vivo, preclinical manner that previously could not be studied.
This model could potentially be used to study diseases such as glaucoma, traumatic brain injury, idiopathic intracranial hypertension, and spaceflight-associated neuroocular syndrome.
To set up the inflow syringes, load a 30 milliliter syringe with 30 milliliters of the perfusion fluid of interest and attach a three-way stopcock to the syringe. Attach a 0.22 micrometer hydrophilic filter to the stopcock and attach a 15-gauge lure stub adapter to the 0.22 micrometer hydrophilic filter. After removing air bubbles from the syringe setup, attach tubing to the lure stub adapter and close the side port of the stopcock with an unvented universal lock cap. After setting up a second inflow syringe as just demonstrated, label one syringe channel 1 ICP and label the other syringe channel 2 IOP. Then set up the outflow syringes as just demonstrated, but without loading the syringes with the infusion medium. To prepare a human whole eye globe sample, remove the optic nerve sheath and remove the vitreous humor from the posterior segment. To ensure a good fit on the round dome of the IOP chamber,